All right, as we learned in the first learning objective, there are two parts to the adaptive immune system. There's the cellular component, which is the T cells, and there's the humoral component, which are the B cells. Now, the book did not provide a learning objective for figure 12.2, but it is a very important figure to know. So I've made my own learning objective 1.5, and this will follow along with figure 12.2. There are 4 stages in each of the cellular and humoral responses. The first, excuse me, that was a wet cough, not a dry cough. The first step in the adaptive immune system is called antigen presentation. This means that you have to sort of show the T cells and the B cells the different foreign particles that are being found in the body. In order for the T cells to be activated, they have to have an interaction with an APC cell. This is an antigen presenting cell. They are required to interact with this APC cell. Typically, these are dendritic cells. B cells, on the other hand, they can sort of find antigens on their own. So as you can see in the picture, you can see that the T cell is associated with the APC cell. The B cells, they can interact with the APC cell, but they're not required to interact with them. They can find antigens on their own. All right, stage 1. We need to find foreign particles, because until we find foreign particles or antigens, there's no point in us being around. Next, once we find one, we need to get activated. So step 2 is called leukocyte activation. You'll notice that once these cells have found a foreign particle and antigen, they are then going to release cytokines, just like we talked about. They're part of the second line defenses, but they're also part of our third line defenses. Chemicals are going to help us to communicate. These cytokines will then be able to activate. So for both of these, cytokines will activate both of these types of cells. So what does it mean to become activated? That moves us into stage 3. To become activated means that we will proliferate and differentiate. So stage 3 is proliferation and differentiation. So proliferation, what you notice in the picture is we started off with 1 cell, now we have 3. So proliferation is just another word for mitosis. Remember, these are eukaryotic cells. You are a eukaryote. So to undergo proliferation means we will undergo mitosis and we will make more of our cells. After we've made new cells, we will then begin to differentiate. T cells will take 2 main pathways. T cells can develop into memory cells, or they can become effector cells. We'll talk a little bit more. Effector cells, we'll talk about a lot of different types of effector cells. Memory cells, once they differentiate, they're going to go into the lymph system so that they can be that memory. Remember, the lymph nodes in the spleen are going to filter the blood and the lymph. It's these memory cells that are going to be looking for particles as they go through there. Effector cells are going to be what will go to those sites of infection and be able to eliminate and get rid of our pathogen. B cells, they can also undergo mitosis, and as they differentiate, they will also form memory cells. So both T and B cells will differentiate into memory cells, but B cells, they will differentiate into what are called plasma cells. These are the equivalent of effector cells. These are the ones that will create antibodies. So you've been hearing a lot in the news lately about antibodies associated with COVID-19. What they're finding is these antibodies are proteins that are made by these plasma cells in order to tag the different pathogens. Remember our complement proteins? In the classical pathway, they would look for these antibodies. Well, we can also look for antibodies in a person to determine if they have been affected by a disease, and that's how we can know if someone has had a disease even if they've never been tested for it. So as you hear people being tested for antibodies, this is what they're looking for, and they would come from our B cells. So finally, we have our different types of cells that can fight our incoming pathogen. And so the last stage is they have to go do their job, which is either elimination of the antigen, or they will go and reside as memory cells. Remember the role of the memory cells. These are what are going to be helping us to filter the lymph and the blood in the spleen and in our lymph nodes. The effector cells, those are the ones that actually go to the site and actually will fight the infection. So, elimination, this is what is done by those effector cells and those plasma cells. These will both actually fight the infection. So once they are done, remember during resolution, once they're done, these are the ones that will eventually kill themselves after they're done with their job, and they will become part of that pus. So those are the 4 stages of adaptive immunity.